Hello everyone, I am Dr. Fatima Oyelowo-Abdulraheem from the Department of Veterinary Anatomy, Faculty of Veterinary Medicine, University of Abuja, Nigeria. I'll be presenting fluorescence microscopy, which is a part of the introductory series in microscopy training. At the end of this presentation, you should be able to understand the history of fluorescence, describe how fluorescence occurs, the types of compounds that undergo fluorescence, the concepts associated with fluorescence, the characteristics of fluorescent dyes, and some of the challenges associated with fluorescent probes. Why fluorescence? Why do you choose fluorescence over other types of microscopic modalities? This is mainly because it is used to differentiate between objects. It's an important tool for observing the distribution of specific molecules. What if you want to visualize objects that don't provide enough contrast? Although bright field stains give better contrast through color but have limited specificity, fluorescence can give excellent contrast and specificity. Take a look at these images of phase contrast and differential interface contrast, DIC. It's quite different from fluorescence. You can see the nucleus is much more clearer in beautiful colors. The benefits of fluorescent microscopy are high contrast, high specificity, high sensitivity, high stability, high quantitativeness, and live cell imaging. Take a look at this image. It's multicolor nucleus with fluorescent image showing labels to differentiate and demarcate the cellular organs, such as nucleus and cytosol, including the components such as actin and microtubules. This image cannot be seen on phase contrast or DIC. So let us start with a brief history about fluorescence and how it came about. It was first described as early as 1500s for certain three derivatives. It was studied in the 1800s for chlorophyll and quinine. Both chlorophyll and quinine are important biological molecules. Fluorescence is the emission of light by a substance caused by the absorption of radiation at a specific wavelength, followed by nearly immediate emission at a different wavelength and longer wavelengths. First, typically, observation of fluorescence was of a molecule called quinine. It's a drug that has probably benefited more people as it combats infectious disease. This was of a great interest in the 19th century as it was used as anti-malaria. John William Herschel first described fluorescence about over 200 years ago. Under certain incidences of light, quinine was seen as vivid and a beautiful celestial blue color, which from the circumstance of its occurrence would seem to penetrate in those strata in in which light first penetrates. He was a mathematician, a chemist, and a photographer. Sir George Stokes explored fluorescence in a more systematic way. He introduced the idea of filters tied with the electromagnetic spectrum, showing how the wavelength is associated with various fluorescent molecules. Here he used a stained glass as a filter. The glass absorbed all the light greater than 400 nanometers to allow only UV light to enter a sample. The tonic water was used and a colored goblet was also used to allow light to pass through. In this way, he isolated the blue color associated with quinine. Fluorescence also associated with minerals, that is the fluorspar. Over the next 200 years, enormous growth in this discipline was seen, and fluorescence is a very important aspect of microscopy today, especially as regards to biological samples. What is fluorescence? Fluorescence is a physical process whereby light is absorbed by a molecule and light of lower energy is released. When we shine a blue light onto fluorescent, the molecule gives off a green light. A fluorophore is a chemical compound that emits light upon excitation. It typically contains several aromatic groups or cyclic molecules with pi bonds. In this case, the chemical structure is fluorescent, an important compound in the history of fluorescence and in microscopy today. The process of fluorescence consists of 1. Absorption of light, the absorption of a photon that excites an electron. Secondly, once the photon has absorbed the light, the molecule absorbs specific wavelength and almost immediately releases photon with lower energy. So the emission is the part of the second process. Remember that wavelength of an emitted light is always longer than that of the light that is absorbed. This is a decrease in energy as the wavelength of the light emitted has a longer wavelength. Why does fluorescence not fluoresce if we shine an orange or red light to it? This is because specific wavelengths of lights are required to cause fluorescence. Why does the fluorophore require specific wavelength of light to fluoresce? The illumination light, also called excitation light, must have enough energy, and that takes us to Jablonski diagrams. We can use a Jablonski diagrams to understand how fluorescence works. It's a representation of a molecule's electronic states and the transitions between them. moving is the diagram typically shows us the possible energy, that is the possible energy states of the electrons in a molecule. Moving upward represents a greater amount of energy. When a molecule absorbs a photon, it gains the energy of that photon. If the composition or structure of that molecule is appropriate, some of that energy goes to the electrons. If there is enough energy, the electron will be more or become excited and move from lower energy, that is the ground state, which is the SO, into a higher orbital energy, which is the excited state, that is S1. Once it is in this excited state, the electron can lose a small amount of energy. You understand? Once it loses a small amount of energy, it settles briefly on the lowest vibration level of this state, that is the bold line. The energy is lost in form of heat, that is the vibration. Almost immediately, the energy that has been lost will now settle and will be reduced at the S0 state. What can fluorescence? A lot of compound can undergo fluorescence. One is laundry detergent. It contains blue fluorophores that make white clothes appear brighter, giving off blue light. What color light is required to excite a blue fluorophore? We also have special rock materials such as opal, also fluorescence under UV light. So does quinine in tonic water. Flowers also fluorescence to attract insects. If so many things can fluorescence, why do we need special fluorescent dyes? Because there are a number of characteristics that describe a molecule's ability to fluorescence. We want to use these molecules that make it easier to fluorescence. And that brings us to the characteristics of fluorescence molecules. And these good characteristics are bright color, specific color. There are also differences in excitation and emission energy. Fluorophores can absorb and emit light of varying wavelengths. The energy that is released as heat by electron can vary. This means that the electron will produce light of different wavelengths as it relaxes. So the energy is released when the energy released is less than that energy that was absorbed. So as we see this, at the end of the day, you realize that if you look at this wavelength, the energy that was absorbed at the end of the day, the energy is lost as heat because of vibration. If we combine all the possibilities of light that can excite a fluorophore and combine all the wavelengths of light that is emitted, we can then produce a spectrum. This is a plot of excitation and emission spectra of fluorescent. Each plot can be interpreted as representing the probabilities of excitation or emission occurring respectively. Therefore, fluorescence can be excited by 450 nanometers or 500 nanometers. Light, for example. Taking a look at this image, the likelihood of fluorescence occurring at 450 nanometers is approximately half of that of 500 nanometers. Therefore, twice as much light is required to produce the same fluorescent intensity at 450 nanometers than 500 nanometers excitation. We obviously do not want to waste light when looking at samples. Therefore, the fluoroprobe will be most efficient, or rather will be most efficiently excited using light at the peak of the spectrum. That is, you can see the excitation spectrum of 450 and the emission spectrum. One excitation photon is seen at exactly 450. The difference between the excitation and emission peak is referred to as Stokes shift. That is, the excitation peak is the difference to that of emission peak is called Stokes shift. One of the important properties of fluorophore is the excitation and emission peaks. This will be very important when using microscope to detect signal. You have to be careful of spillover in a multicolor experiment. Spillover is a process in which a fluorescent signal aimed to detect only in a given detection channel is also detected in adjacent detection channel. For example, you'll be able to distinguish between the blue and green fluorescence signals shown in this image. You can see an overlap of colors causing a spillover. Note that using these blue and green fluorophores together in the same multicolor imaging experiment is not advisable. The next set of slides will give more insight in this process. If you look at these slides, if 10 photos are used to excite fluorescence, will 10 photons be emitted at the end of the day? Will 10 photons come out? For example, if we now have 2 photons not absorbed, then it means at the end of the day, we'll have 8 photons out. A factor we need to consider is the ability of the molecule to absorb and excite light. So if 2 photons are not absorbed from the 10 photons, then at the end of the day, do we have 8? What if 3 photons do not also result in fluorescence? So we really need to consider if all of the absorbed photons result in fluorescence. So if 2 was not absorbed and 3 do not result in fluorescence, then at the end of the day, we might have 5 photons out. energy from the photons will also be lost to other processes, such as heat. Light is also a very important factor to consider when performing fluorescence microscopy on biological samples. We do not want to expose the sample to too much light, as it will eventually damage the sample. So what are these processes called? So we have the quantum yield, which is the or we take the extinction coefficient first. The extinction coefficient is the capacity for light absorption at specific wavelength, and it is measured in 1 centimeter, while the quantum yield is the ratio of the absorbed and emitted photons, that is, how much is emitted. The brightness at the end of the day released is the output of a molecule. It is equal to the product of both the quantum yield and the extinction coefficient. If you look at this table, you will see the quantum yield. You also see the extinction coefficient. Don't forget that the quantum yield is the ratio of absorbed and emitted photons, and the extinction coefficient is the capacity for light absorption at specific wavelength. The higher the extinction coefficient, the more likely the compound will be absorbed, the more likely the photons will absorb light. The quantum yield is a number between 0 and 1, giving an idea of how many absorbed photons will contribute to the fluorescence. Brightness is the product of the two, that is, product of the extinction coefficient and quantum yield divided by 1000. Thus, the more the brightness, the more efficient it is in converting light into fluorescence. What happens when an absorbed photon does not result in fluorescence? If the quantum yield is not 1, photons are lost along the way. Some are lost as heat, and that takes us to the dark side of fluorescence. In addition to producing heat, an excited electron can sometimes result in processes other than fluorescence, that is, non-radiation decay. A photon can undergo a process of intersystem crossing, move to an excited triplet state, then relax to the lowest vibrational level, giving a reductive decay, and return to ground state. So this leads us to what you call photobleaching, and what causes this photobleaching. Don't forget that in the excited triplet state, the fluorophore interacts with molecules around it, usually oxygen, causing a reaction. The molecule is chemically changed and electron is now lost. Thus, the fluorescent molecule can no longer be excited, and that is what is referred to as photobleaching. That is, photobleaching has occurred. The electron can then interact with the molecule around it, such as molecular oxygen, which causes a reaction, producing reactive oxygen species, that is the ROS. Now, photobleaching is a permanent loss of electrons from the productive cycle of fluorescent emission due to photochemical change. Characteristic decay when measuring a large population of fluoropores. Usually it is an irreversible mono-exponential process. On the other hand, photostability is very important when designing your experiment. A molecule that is less likely to enter the triplet state is less likely to become photobleached. But there are other factors too. Use of photostable dyes. For you to have a photostable dyes, you need to make sure that you have projected your photons. You only radiate your specimen when acquiring. You also reduce laser power or exposure time. You anti-fade reagents that can help by removing the ROS, that's reactive oxygen scavengers. Other blocking media can also be used. You can also have aqueous or glycerol plastic based, and hard or soft settling or concentrated liquid, and you also label densely. Photostability of different molecules. The more photostable the molecule, the better for you. How to prevent photobleaching. When a fluoropore loses its fluorescence due to damage induced by light, you have to avoid this. And the practical advice on how to avoid photobleaching are staining in a semi-dark conditions, keep samples in dark containers or wrapped foil, short excitation or exposure time while imaging, reduce excitation power as low as possible. This brings us to types of fluoropores. We have three types of fluoropores. The fluorescent dyes, which are small, the fluorescent proteins, which are large proteins, and the quantum dots, which are nanoparticles. We take the first one, the fluorescent dyes. These are small molecules, and they come in families such as xanthine, cyanine. These photos shows a whole spectrum of colors represented by the fluorescent dyes. The properties of these are that they can often tell us the interesting aspect of their environment. They are very sensitive to the environment. They can act differently in different environments, like high or low pH, They can also give ionic concentrations of calcium, magnesium, and sodium. They also have lipid versus aqueous surroundings. Can also have temperatures and viscosities. This image shows a near red under visible and UV light in different solvents and pH levels. Characteristics of fluorescent dyes: 1. Selectivity. 2. Signal to background. 3. Photostability. 4. Excitation or emission properties. And 5. Environmental stability. The second class is the fluorescent proteins. They are the large molecules and first described in jellyfish. It produces flashes of blue light by a quick release of calcium, which interacts with photoproteins aquarium. The blue light is produced, in turn is transduced to green by the now famous green fluorescent protein. The fluorescent protein, which are the large molecules, does need to be genetically encoded into the biological system. This diagram shows a wide range of spectrum of fluorescence produced by proteins. You can see the cherry, you can see the orange, you can see the tomato, you can see the strawberry or banana. And we now come to the third group of nanoparticles, also called quantum dots. They are nanocrystals of certain semiconductors and are highly fluorescent. Their crystals are low nanometer size and they must be coated with a biocompatible material such as lipid or PEG. Fluorescence labeling. Fluorescence labeling are done by different methods. First, we have the chemical labeling. Here, the chemical is done chemically through a covalent binding of fluorochrome to the protein of interest. Secondly, you have genetically. Here, fluorescent proteins are genetically encoded, and you can have immunolabeling. Here, antibodies are going to be labeled with a fluorescent. This also brings us to fish, which is fluorescence in situ hybridization, a laboratory technique for detecting and locating a specific DNA sequence on a chromosome. Although there are several advantages of fluorescence, there are still several challenges associated with fluorescent probes. We have photobleaching or phototoxicity, have autofluorescence, spectral overlap, as you've seen in previous slides, sample preparation, and lastly, labeling phototoxicity. Now, fluorescence microscopy itself. We'll talk about it actually on the surface. It has components and concentrations for microscopy, which is similar to other microscopy. The components are light source, collimators, polarizers, filters, beam splitters, detectors, and you also have to take the resolution into consideration and the contrast. Fluorescence microscopy modalities. We have wide field fluorescence microscopy, confocal, total internal reflection, light sheet fluorescence microscopy, and two photon fluorescence microscopy. We're going to talk about the wide field fluorescence microscopy, while the others will be described at the intermediate level of the curriculum. So, wide field fluorescence microscopy. If you take a look at these two images, you see the tissue section and that of the monolayer of cells. If both of these images were taken with the wide field fluorescence microscopy or fluorescent microscope, then why is it the tissue section clearer than that of the monolayer of cells? If they are both roughly the same thickness, why is there difference in the clarity? In one case, all axial information is lost, and in the other, the images are blurry and we cannot see the detail, that is the low contrast. Wide field fluorescence does not provide good axial information as light originating outside of the focal plane will cause the image to be blurry, corrupting the image. This is because out of focus light still reaches the detector. It is important to discriminate light from different focal planes. What microscope do we need to use to restrict the light to that from only one focal plane? You can slice the sample very thinly, but that can compromise the integrity and is obviously incompatible with live sample. The tissue section is imaged with a much lower numerical aperture and magnification. Therefore, the depth of the field is much larger than the case of the monolayer of cells. But in both cases, we are seeing structures and fluorescence from more than one focal plane. In a microscope, light from different focal planes will be focused differently. This is most notable with high numerical aperture objectives, precisely when you want the best clarity. In many cases, it is not ideal to observe the extra information from other focal planes, as it will skew our interpretation of an image. Why use wide field microscopy? Advantages: it is simple and very versatile. The disadvantages is that it has no axial information and it's not useful with thick samples. Take a look at this image. From your end, you'll be seeing it as if the girl has the head of a horse, because from this side, from this axis, you can see it as if the horse and the girl are one object. But on the other side, it can be seen that the girl is sitting close to the horse, and thus the horse and the girl are different entities entirely. This is a schematic diagram of a simple wide field microscope. You can see the excitation light passing through the first optical filter. It is focused on the spectrum from above. Fluorescence is then directed to the detector after passing through the emission filter that rejects and reflected and scatters excitation light. So, what is the difference between a wide field microscopy and a confocal microscopy? Looking at these two images, confocal microscopy can limit detection to only one light, as in limiting the light into the focal plane of interest. This is done using a pin hole. Getting rid of the light originating outside of the focal plane removes the focus light, making your sample sharper and clearer, as seen in this image. Thank you, and the floor is now open for questions.